So, welcome to the third day of the Computational and Experimental Chemistry Symposium. The symposium is dedicated to bringing together the computational simulation community and experimental researchers to exchange scientific ideas and to learn the current state-of-the-art modeling, analytical, and numerical tools together with advanced experimental techniques. We hope that our attendees, colleagues, and their families are safe in the ongoing COVID-19 pandemic. Today, there will be two lectures presented by Dmitry Lapshin from the University of Edinburgh and Dr. Breanndan O Conchuir from IBM Research. I'm Dmitry Lapshin, a PhD candidate at the University of Edinburgh and initiator of this series of lectures. Questions to our speakers from Zoom and YouTube will be moderated by me. Please use chats to ask questions at any time. I would like to acknowledge technical support provided by the International Adsorption Society and Dr. Dan Siderius in particular. Now I would like to introduce the first speaker, Dmitry Lapshin, and that's me. So let me share my screen. So as I mentioned, I'm a PhD student in chemical engineering at the University of Edinburgh. My research interests include molecular simulations and experiments devoted to understanding of interfacial processes in application to gas adsorption and surface characterization. Some of my contributions, I contributed to an understanding of gas adsorption in ionic liquid films, and that's the topic of my talk today. And I also contributed to patents on application of mechanochemistry in the field of fire safety. So today I would like to discuss a relation between gas adsorption on the surface of liquids and gas absorption in bulk regions of liquids. And the framework of my talk falls within mostly gas adsorption and separation, for example, carbon dioxide from industrial gas emissions. However, computational methods and approaches that I'm going to discuss today, they can be applied to other fields of research where interfacial interactions take place. For example, adsorption of gases in liquids, heterogeneous catalysis processes associated with phase separation, extraction, and hydrophobicity. Thus, I will specifically focus on interfacial interactions between gas and liquids. I would like to split my talk into two parts. So in the first part of my talk, I will introduce the main area of my research, and I will try to answer the following questions. What kind of simulations do we need in order to model gas-liquid interfaces? Which computational tools and approaches can be used in order to study gas adsorption properties of liquids, and how these computational methods can be related to experiments. I will consider different methods and theories, and I will compare them, and I will try to find the best one that provides quantitative characterization of gas adsorption in thin films of liquids. And then in the second part of my talk, I will talk about the benefits of using simulations together with experiments, and how using both computational and experimental methods we can predict gas adsorption in liquids. To answer the proposed questions, I will use my research as an illustration, which is a collaborative work together with my supervisors, Professors Lev Sarkisov and Eleanor Campbell, and my colleagues, Doctors Miguel Josh and Andrei Groma. So nowadays, one of the most mature and well-established technologies for selective carbon capture is based on ComECS processes, and in particular on application of amine-based solutions. This technology suffers from low mass transport and high energy demand due to the recovery of the solvent. And on the other hand, physisorption processes in porous materials, they offer fast mass transfer and large exchange surface area. However, they don't have chemical specificity of amines. So the combination of these both technologies, in particular, if we have a porous material like here, which have a film of an active liquid on its surface, so this material has an advantage of high mass transfer and also large interfacial area. And in this particular research, we are interested in depositing ionic liquids on the surface of this porous material. Ionic liquids have been considered as promising solvents for gases because they have negligible vapor pressure that solves the problem of recovering the solvent down the stream of the regeneration process. Before I go to the discussion of adsorption properties in thin films and establishing connections between simulations and experiments, I would like to outline which kind of information we can extract from experiments, because I will use this information further in developing a predictive model of gas adsorption in liquids. So imagine the following situation. We have a material that looks like this. So we have a thin film of ionic liquid deposited on the surface of a porous material. And from experiments, we can measure the surface area of this material, its pore volume. We also know temperature, gas composition, or pressure. We can assume that the liquid phase is uniformly distributed inside pores. And essentially the question that we need to answer is, what can we say about adsorption in this material? So if we look at this material, we intuitively can see that some of the properties of this material will depend on the interior of the film, and these processes are associated with the gas solubility. And some properties of this material will depend on the surface, and in this case we deal with gas adsorption. And we also have interactions between the solid and the liquid phase. However, we are not going to consider these particular interactions today. For bulk regions, such as bulk liquid phase and bulk gas phase, we have experimental tools in order to measure, for example, gas solubility in liquids and pressure in a free pore volume. But what is really difficult to study in experiments is the interface, and that's where computational simulations can provide us with additional insights. So at the heart of all computational methods and simulations that I'm going to discuss today, there will be a system that looks like this. So we have a simulation sample which consists of a film of ionic liquid directly exposed to the gas phase containing carbon dioxide and nitrogen. We specifically focus on this particular ionic liquid, which has this chemical composition and structure. Because this is a well-studied ionic liquid, so we can relate our results to reference studies. We start our simulations with random distribution of gas molecules in free space, and then just let the system equilibrate. So we run long molecular dynamics simulations in a canonical ensemble where the system has the same number of particles that occupy a certain volume at a particular temperature. All molecular simulation details are taken from this paper, and simulation details can be found here using this link. So how can we relate these molecular dynamic simulations to experiments? A canonical ensemble is analogous to experimental conditions. On the left, I'm showing a microsystem that I'm simulating, and these simulations, they provide velocities and coordinates of all particles present in this microsystem. And using these velocities and coordinates, we can calculate average thermodynamic properties of a macro system shown on the right, such as pressure, temperature, density, solubility of gases in the liquid phase. And in this particular research, the key property that we are interested in is density of gas adsorbed on the surface, dissolved in the bulk liquid phase, and present in bulk gas phase. So once we have an equilibrated system, we can now apply structure characterization tools. We have two methods to look at the structure shown here. Normally, and this is what has been used by different research groups, you calculate a density profile of liquid and gas by simply dividing the system into slabs and counting the number of particles within each slab. And this is a typical density profile you would get. So we can recognize bulk liquid phase, vacuum phase, for example, or gas phase, and a transition region associated with the interface. So let's call this profile global. In my work, I use a method that was extended by Dr. Miguel Josh, and this method recognizes the very complex structure of the interface. So the method actually traces the shape of the film. We also split the system into slabs and count the number of particles with respect to this complicated surface. And this picture shows how the ordering is recognized by this method. So if we look at this density profile, these two peaks are associated with a very ordered structure of the surface layer. Then we have essentially nothing here, so we have this minimum, and two additional spikes are associated with a less ordered subsurface layer. And then we have a uniform bulk density associated with just random distribution of particles in the bulk liquid phase. So if you have an experimental system which deals with for example, gas-liquid interfaces or liquid-liquid interfaces, then you can do simulations and apply the ITM analysis in order to understand the structure of the interface. So this method can be applied to any fluid-fluid interfaces. Additional points that I would like to make are the following. So the intrinsic density profile indicates the actual location of gas molecules adsorbed on the surface in comparison to the global density profile. Moreover, the ITM method, it also allows us for the correct calculation of the excess amount of gas adsorbed relative to the intrinsic surface. In the global density profile, we have to use Gibbs dividing surface, and if we compare these areas below the solutes curves, there will be a difference, significant difference between them, especially for the for blue areas. So at this point, I would like to finish my first part, and here I provide some useful references on the theory of the intrinsic analysis. And if you have questions, I can answer them. So let me. So let me see if we have some questions. So we have a few questions. The first question comes from YouTube. It's asked by Mindert van der Spek. The question is, how can you assume that the liquid is uniformly distributed? Is that an educated guess, or were you able to characterize that? So I guess this question is related to the distribution of ionic liquid in porous material. So honestly, this is just an assumption that we just assume that the liquid is uniformly distributed inside these pores. However, in reality, we don't really know whether ionic liquids are uniformly distributed inside pores, or maybe they just fill pores or create some distinct droplets on the surface of pores. So some experimental studies, they say that ionic liquids are uniformly distributed inside, for example, porous silicas or carbon. But this is still a question, I would say. The second question comes from Zoom, from Pierre Gisquire. Can this ITEAM technique be applied to porous amorphous materials? So if you have, I think, just a porous material and gas, you can apply this method to these systems, but I think you will get the same picture. Let me just go to my presentation. So if you have porous material and gas, you can apply the ITEAM analysis. However, I think you will have the same density profile as global density profile. So in this case, so for example, in my research, I use GROMACS software that allows to calculate these global density profiles. And in this case, I would use these global density profiles and just GROMACS in order to calculate these profiles, because well, the ITEAM method, you still need to do some additional calculations. The ITEAM method, it's beneficial if you have liquids where the surface is non-uniform, so it's perturbed. And in this case, in order to catch this complex surface, in this case, you apply the ITEAM method. So I hope I was able to answer this question. So now I would like to go to the second part of my talk and illustrate how simulations and experiments can support each other. But before this, I would like to focus on the ITEAM density profile of the liquid phase on the black curve. Just to illustrate what can we learn from the iTeam analysis. So I will go briefly through this patch because the main subject of my talk is gas adsorption. So these spikes, as I mentioned before, they are associated with a specific alignment of ions on the surface and subsurface layer. And using number density profiles of atoms, we can calculate a population of atoms that are essentially on the surface. So the surface, from this picture, we see that the surface is mostly populated with these CT atoms associated with terminal methyl groups attached to the butyl chain. This suggests that the cation mostly orients its butyl chains to the gas phase. We also find some amount of methyl groups attached to the imidazolium ring, and this means that we have another orientation mode where the imidazolium rings are present on the surface. And we also see that there is a notable amount of anions on the surface. So this is just some illustration, a very brief illustration of what we can say about the structure of the surface layer. Of course, you can apply some other analytical techniques in order to extract more information. So now I would like to move to the analysis of gas density profiles, and I will focus on the CO2 profile. First of all, the intrinsic surface allows us to delineate the liquid, clearly delineate the liquid phase and the gas phase. So we can use this intrinsic surface in order to calculate the amount of gas dissolved in the liquid phase, the amount of gas present in the bulk gas, and the excess amount of gas adsorbed. So actually we see that the surface acts as an attractive surface, and this causes the presence of this excess gas amount adsorbed. We see that one part of the system is governed by the properties of the liquid phase, and another part of the system is governed by the interfacial properties. So in order to predict the total amount of gas adsorbed in this system, we need to calculate all these three contributions. And then we just combine them and find the total amount of gas adsorbed, or we can predict this quantity. What is also important to say is that now we can actually find a region of the bulk ionic liquid which actually acts as bulk liquid and does not influence and does not feel the influence of the interface. So in this picture, I'm showing CO2 absorption isotherm for ionic liquid, and this picture tells us two things. The first thing is that the liquid phase definitely behaves as bulk ionic region, and also computational simulations can reproduce experimental CO2 solubility in this ionic liquid. So in order to develop a predictive model, we need to introduce a parameter that actually characterizes the competition between gas adsorption on the surface and gas absorption in bulk liquid phase, or essentially competition between yellow and red regions. So why is it important? If we have a material with a film of ionic liquid deposited on the surface, and as we increase the thickness of the film, this material will behave more and more like bulk liquid. And we need to find such a metric that given experimental data such as surface area per volume, the amount of ionic liquid that we add or put inside pores, that can characterize this competition between gas adsorbed and gas absorbed in the bulk liquid phase. And this parameter, it should be also a relation between these two quantities. So we call this parameter the equipartition thickness, and this is the thickness of ionic liquid or the liquid phase when the amount of gas adsorbed on the surface is equal to the amount of gas dissolved in the liquid phase. So as I mentioned, this is a relation between yellow and red densities of gas. Or if we are talking about Henry's law regime of adsorption, then we can relate these densities to Henry's constants and find the equipartition thickness as the relation between these two Henry's constants. This is a useful quantity because it's constant for the case of low pressure adsorption or the Henry's law regime of adsorption. So we use this equation. However, here we see that for nitrogen, the equipartition thickness changes as a function of pressure. And we have to apply this equation, and that's because gas adsorption of nitrogen on the surface of ionic liquid does not follow the Henry's law regime of adsorption. So, if we are talking about low pressure adsorption, we can use this constant to predict the total amount of gas adsorbed. So let me now illustrate how using the equipartition thickness and, as I promised, using the initial experimental data such as the surface area per volume for the pristine porous material and the material that contains some amount of ionic liquid. We also should know pressure and gas composition. So how, using all this data, we can predict the total amount of gas adsorbed. So gas solubility in bulk liquid region we can measure from experiments. So we can obtain the Henry's constant from the experiment. Using an equation of state, we can calculate the density of gas present in free pore volume, and then using one simulation, from one simulation we calculate the equipartition thickness, and since it's constant, then we can just use it for any pressure where the Henry's law applies. And combining this equipartition thickness with experimentally available data, we can calculate the excess amount of gas adsorbed. So we have all three contributions and therefore can predict the total amount of gas adsorbed in this system. Using the same strategy, we can uh, investigate how selectivity changes as a function of thickness of the film. So here I'm showing actually uh, selectivity of CO2 over nitrogen as a function of thickness, and we see that the surface has low selectivity of 5, while the bulk region of ionic liquid has large selectivity of 83. The black curve is the ideal selectivity, where we used Henry's constants, and the red curve, it's calculated for a more realistic system, a flue gas, which contains 15% of CO2 and 85% of nitrogen. So in any case, we see that the presence of the interface lowers selectivity for any reasonable thickness of the film. So now I would like to conclude, to make some conclusions. So molecular dynamics simulations, they provide additional insights into gas adsorption in thin films of ionic liquids that are not available from experiments. And this is related to the interfacial properties. The ITM method should be used for quantitatively accurate characterization of interfacial properties. Combining both simulations and experiments, we can predict the total amount of gas adsorbed in films. And this is possible if we introduce the equipartition thickness, but only for the Henry's law regime of adsorption. Thin films of ionic liquids have high gas capacity, but low selectivity makes them inappropriate for gas separation. And finally, gas selectivity of liquid films has two limits. The interface has low selectivity, while the bulk liquid region of the film has high selectivity. So using the equipartition thickness, we can not only to predict the total amount of gas adsorbed in films, but also to design task-specific materials with different contributions of surface and bulk properties. So here I'm providing some additional references to gas-liquid interfaces, and my talk is based on our recent paper. And again, our listeners can find simulation details and methodology on the ITM method here using this link. So if you have questions related either to the first or second part of my talk, I'll be happy to answer them. And thank you for your attention. So now I would like to introduce the next speaker, Dr. Breanndan O Conchuir. So Breanndan is a researcher, staff member at IBM Research Europe in the UK. His research interests include data-driven methods and particle-based chemical simulations in the areas of rheology and surface science. He established several collaborations between industry and academia in the field of modeling of bioprocesses and complex nanoparticles, and today he will speak about solid-liquid interfaces. So now I would like to give a word to Dr. Breanndan O Conchuir.
So the title of my talk today is The Role of Chemical Heterogeneity on Surfactant Absorption at Solid-Liquid Interfaces. And the work which I'm about to show you was produced as part of a collaboration between the Science Technology Facilities Council and IBM Research. And this collaboration was funded by the Department of Business, Energy and Industrial Strategy. So first of all, what exactly is a surfactant? Well, a surfactant is an amphiphilic molecule where one end of the molecule is composed of functional groups which are hydrophilic and are attracted to water, while the other end of the molecule is composed of functional groups which are hydrophobic and are repelled by the water. And typically surfactants are added in order to augment the interfacial tension between two liquids, between a liquid and a gas, and between a liquid and a solid. So in this schematic here, we see several surfactants migrating or absorbing at the water-oil interface to help stabilize an oil droplet. So why are we interested in the absorption of surfactant onto solid surfaces? Well, the reason for this is because this phenomenon is ubiquitous in both nature and many industrial processes. So take, for example, cleaning products or detergents. So here, surfactants are added to the liquid and migrate to the interface between fatty solids and liquids to change the interfacial tension to allow the water to penetrate into the stain and into the fatty deposit in order to sweep away small droplets of grease and dirt particles. However, sometimes the absorption of surfactants onto solid surfaces disrupts an industrial process and the overall performance and efficiency of these processes. So take, for example, enhanced oil recovery. So here, in many cases, surfactants are added to aqueous solutions where they move to the interface between the water and the oil to change the interfacial tension to facilitate the extraction of more oil deposits from the reservoir. However, the surfactants can also absorb onto nanoporous rocks in the reservoir, reducing the overall concentration of surfactants at this water-oil interface and the overall effectiveness of this process. And similarly, for applications such as lubricants and engine oils, the absorption of surfactants onto solid surfaces is fundamental to its operation. So here, surfactants are added to a base oil that then absorbs onto the surface of inorganic surfaces or inorganic materials to produce a protective layer which reduces the overall friction in the engine. And this phenomenon of surfactant absorption on solid surfaces is also fundamental to the micellar polymerization. So here in the top schematic, you can see, sorry, just in the top schematic, you can see that an admicelle is formed, absorbed by a layer of surfactant on the solid substrate. And then monomers are localized within this bilayer. They then become polymerized, and then the upper layer of surfactant is washed away. And similarly for coatings, we have surfactants that become absorbed onto metallic surfaces in order to inhibit corrosion and fouling. And finally, in the application case of nanoparticle stabilization, we see surfactants which absorb onto the surface of colloidal particles. They then repel one another via this steric repulsion, which prevents them from aggregating with one another and helps maintain colloidal stability in the dispersion. And there are many other examples and instances where the absorption of surfactants onto solid surfaces is important or fundamental to industrial applications and processes, but I won't go into any more at the moment. Now, the fundamental problem which one encounters when one wishes to study the absorption of surfactants onto solid surfaces is that in many cases, or in most cases, these surfaces tend to be rough, and they also tend to be chemically heterogeneous. And we have seen in the literature cases where these two effects can change the number of absorbed surfactants. It can disrupt the profile of absorption isotherms, even the heat of absorption, and the morphologies of the absorbed surfactant assemblies or micelles themselves. And of course, our lack of understanding of how this happens inhibits our ability to produce better industrial applications and processes similar to the slides which I've shown you previously. So in this work, we interrogated the interplay between cohesive micelle forces, cooperative effects, and surface interactions, and investigate how all of these components affect the overall macroscopic absorption characteristics of the surfactants absorbing on chemically heterogeneous surfaces. And with this understanding, we developed and demonstrated, we can develop and demonstrate a general computational framework for the design of chemically heterogeneous nanopatterned surfaces for bespoke or optimal molecular absorption processes and applications. Now, the simulation methodology which we used in this study is called Dissipative particle dynamics, and this is a coarse-grained molecular dynamics simulation methodology. And the reason why we use this methodology is it allows us to access length scales and time scales which are large enough so that we can investigate the formation of micelles and absorbed micelle structures, as well as conserving enough of the chemical information such that the physics of the individual surfactant molecules themselves are conserved. And the reason why I use DPD over perhaps atomic simulations is because in literature we've seen that's been used to investigate the restructuring of surfactants within micelles, to investigate high surfactant concentrations, as well as the cooperative effects between absorbed micelles. So how does dissipative particle dynamics work? Well, it involves, at a base level, the grouping together of one or more atomic or molecular entities to produce these idealized spherical DPD potentials shown here on the bottom of the slide. And these DPD potentials interact with one another via this simplified inter-bead DPD potential, which has three components. We have a conservative force, we have a drag or dissipative force, as well as a random or stochastic force. And looking at the conservative force, we can see that this interaction has two tunable parameters, namely an amplitude prefactor parameter Aij and a cutoff distance or C. Now in this study, we modeled the absorption of 4 non-ionic surfactants, which were composed of 2 DPD bead types. We have the blue bead types, which you can see on the slide here, which were hydrophilic. And these are the head DPD bead types, denoted by H. And then we also have hydrophobic DPD bead types, colored in green and denoted below by the letter T. And with these 4 non-ionic surfactants, we can examine or interrogate the effects of varying the overall hydrophobic-hydrophilic ratio of the molecule, as well as variations in the length of the molecules. And these molecules were suspended in aqueous solution. So we also modeled water in these simulations. So we had 2 water molecules per DPD bead. And by using the well-established DPD protocol of having 3 water DPD beads per unit volume, we can map from DPD unit lengths to a SI units for length. And within the 4 molecules which we simulated, we have radial bonds, a harmonic radial bond between each nearest neighbor in the linear molecules. And we also have another harmonic bond bending potential between each set of 3 neighboring consecutive DPD beads in the molecule. And of course, we also modeled the solid surface in the study. So we have a cubic lattice of frozen DPD beads. Now, these DPD beads interact with all other DPD beads in the simulation within their interaction cutoff, of course, but they are frozen and therefore do not move. And this cubic lattice is 3 layers deep. And at the bottom layer, we have a layer of impermeable DPD beads, which are strongly repulsive and whose primary function is to prevent the penetration of solvent and solute molecules into the solid surface. And next, we have a middle layer, which was assigned hydrophobic and hydrophilic DPD beads as per the surface generation algorithm, which I will describe shortly. And finally, we have a top surface, which uses the same hydrophilic, hydrophobic pattern as encoded in the middle layer. And the reason why we chose 3 DPD layers was we found that this was the thinnest or the minimum number of layers we could use in the solid slab, such that we could ensure that we have an impermeable solid surface. So next we derived a surface generation algorithm, which was a function of 3 input parameters. So we have 2 generative probability variables, px and py, which describe the order or relative disorder of the hydrophilic hydrophobic surface in the x and y axis respectively. And the third input parameter is S tar, and this is the global target hydrophilic hydrophobic area fraction. So for example, if you want a surface which is 80% hydrophobic and 20% hydrophilic, this variable would be 0.8 and 0.2. And using these 3 input parameters, we can generate heterogeneous hydrophilic hydrophobic surface patterns using a stochastic Monte Carlo approach, which I will describe next. So this is how the algorithm works. So we begin with subfigure A, and by assigning the DPD bead type in the very corner, B11. And just for illustration, I'm going to use red and blue for the assignment of the hydrophilic and the hydrophobic bead types on the surface. So to assign the first corner bead, we take a random variable or tar sampled from a uniform distribution between 0 and 1. And if this random variable is above a 0.6, shown in this example in the global target surface area fraction, then we assign it to be blue. And if it's less than 0.6, we assign it to be red. And next, we assign consecutively the first row and the first column bead by bead using the algorithm denoted at the bottom right-hand corner and the conditions contained within them. So here we extract two random variables, or x and or y, from this uniform probability distribution between 0 and 1. And by comparing them to our generative probabilities, px and py, we can assign bead types to each position. And for our top row and for our leftmost column, if any of the nearest neighbors in those conditions is not assigned, we simply sample a random variable, or tar again, and we compare that to the global target surface area fraction, S tar. And then once we have our first row and our first column assigned, we can then sample from these set of 5 conditions encoded within the algorithm, and until we have a full surface which has been generated. And once we have a numerically generated surface, we can compare the number of red and blue beads which have been assigned to our original S tar, or our target global surface area fraction for red and blue beads. And if they're within a certain tolerance, we accept this stochastically generated surface. Otherwise, we change the or redo or iterate the algorithm to produce a different numerically generated surface. So encoded within this algorithm, we have a geometric two-dimensional patch length distribution, where Lx and Ly describe the length of a consecutive sequence of bead types, all of the same, a consecutive set of beads, all of the same bead type. And this two-dimensional patch length distribution can be decomposed into the patch length distribution in the x-axis, in the y-axis, and the third term there on the left-hand side, a stochastic localized correlation term. But as we go to large surfaces and to infinite surfaces, by the central limit theorem, this localized stochastic correlation term, the third term on the right-hand side of the equation on the left, averages out to be 0. And when we also move from a discrete cubic lattice to continuous space, these geometric probability distributions become exponential. And we end up with 2 mutually independent exponential 1D patch length distributions shown on the right-hand side. So at this point, we have 2 classes of probabilities in this algorithm. So on the left-hand side, we have the 2 generative probability parameters which you use in the assignment of the original surface. And this determines whether or not we randomly sample or assign a bead type from the global target surface area fraction, or if we assign a bead type dependent upon or the same as one of its nearest neighbors. But now we also have a new probability, capital P superscript b, and this is the probability that if you have a bead in the surface of type B that its nearest neighbor has the same B type. And this probability is a function of generative probability Px and Py, but also the global target surface area fraction S tau. And using our exponential 1D patch length distributions, we can integrate out the mean domain length, which we find to be equal to the decay length, which is explicitly included in the probability or the exponential patch length distribution. Now, we simulated 3 classes of surfaces in this study. First of all, we had the purely hydrophobic and purely hydrophilic surfaces. And next, we generated 10 different heterogeneous or disordered surfaces, where we had a global target surface area fraction, which was 50% hydrophilic and 50% hydrophobic. And the only difference then between the different surfaces was the 2 input generative probabilities, Px and Py. And finally, we also generated and simulated 8 different striped surfaces, where we have 2 parallel attractive hydrophobic stripes separated by a hydrophilic repulsive domain. Now, before we start performing any DBD simulation, we need to define the set of interactions between all B types in the simulation. That is to say, the set of prefactor Aij values in the conservative interaction between all pairs of DPD beads in simulation. So in order to get the interactions for the water, head, and tail bead interactions, we performed a series of experiments to measure the critical micelle concentration and the mean aggregation number for a set of surfactants from the CNEM family. And by also simulating these macroscopic observables, we're able to fit values for these, for the interactions between the water, tail, and head bead types. And note that all the interactions that all the beads had with the impermeable bead types were very repulsive and set to have an amplitude prefactor value of 70. And that was purely to ensure that they did not penetrate into the solid surface. And you can find out more information about these experiments in the following paper shown on this slide. So as regards our simulation protocol, we performed DBD simulations using a box of size 80 by 40 by 40, in DBD length units, of course, with periodic boundary conditions in the X and Y axes, with the solid surface slab being at the Z equal to 0 position. And we also performed for each molecule and each surface absorption simulations for 4 distinct bulk concentrations of the molecules. And note that we used identical surface and surfactant force field parameters for both the head and bead types within the surfactants and in the surface. And we also used a well-established sliding window weighted linear least squares protocol in order to determine the time step within the simulation at which point we have reached equilibration. And to ensure that from that time frame in the simulation until the end of the simulation, that our observables, that the time series of the variables or properties of interest to us are not trending upwards or downwards, that they are in fact, from a statistical stance, defined to be stationary. We also applied 2 well-known statistical tests for the property of time series stationary. And for each simulation, we ran exactly 4 million time steps. And this was chosen to ensure the number of equilibrated frames for the observables of interest always exceeded at least 5 times the autocorrelation time of the observable time series. So moving on to absorption metrics, we defined a molecule to be absorbed if one or more of its DPG beads was within a cutoff distance of 1.3125 of DPD units from the top layer of the surface slab. Now, this is very much a heuristic cutoff. However, we found that by varying this quantity, that while quantitatively our results may vary, the qualitative trends which we see are relatively consistent. And by measuring the absorption isotherms on the purely hydrophilic and the purely hydrophobic surfaces, we can also derive a baseline prediction from what we believe the adsorption isotherm should be for the heterogeneous disordered surfaces. So here in this equation, we can see we can weight the absorption isotherms on both the purely hydrophilic and by the purely hydrophobic surfaces by the relative, or by the area fraction of the surface that is hydrophilic and that is hydrophobic. So in our disordered surfaces, we had the 50% hydrophilic and 50% hydrophobic, as I mentioned earlier. And that means that our theoretical estimation or prediction from what the absorption isotherm should be on the disordered surfaces is simply the average of the absorption isotherms on the purely hydrophilic plus the purely hydrophobic surfaces. So examining the absorption isotherms for the predominantly hydrophilic surfactants, we can observe in the simulation that we do not see the formation of micelles because the concentrations are far below their critical micelle concentrations. Generally, we don't see cooperative effects in general, and we have a low number of absorbed molecules overall. But when the molecules do absorb onto the surface, they do so individually as opposed to a part of a greater coordinated or cooperative effect or process. And for absorption onto the purely hydrophilic or purely hydrophobic surfaces, denoted here by the lower curve is the purely hydrophilic surface, and the top curve is the purely hydrophobic surface. And what we found is that the simulation data points, which you can see, fit quite well with a Langmuir-like isotherm, which in one sense makes sense because this is what's been theorized in the literature to be the isotherm profile for non-ionic surfactants below their critical micelle concentration. So for each of the simulations which we ran here, we fitted a Langmuir isotherm, the capsule gamma and the capsule K term, in order to produce the lines or curves you see in both of these plots. And because we have the absorption isotherms for the purely hydrophilic and purely hydrophobic simulations, we can also see in the dashed lines our theoretical prediction from what we believe the absorption isotherm should be for the heterogeneous surfaces. But what is quite interesting is when we compare our simulated data points to this prediction, that we see, particularly in the right-hand side for H6T2, a consistent enhancement of the absorption, or the number of absorbed molecules per surface area due to the fact that we have patterning on the surface. So here in this slide, we show for each of the 10 disordered surfaces, the percentage increase in the number of molecules per surface area that we observe to be absorbed in the simulation relative to this theoretical prediction. And we can see as well for the longer H6T2 that we see considerably more or considerably enhancement or amplification of this increase relative to the shorter H3T1. And when we then investigate the spatial distribution of the density of absorbed molecules, we can see, as we might expect, that they typically absorb onto the attractive hydrophobic domains, shown here in the dashed regions. However, we do see around the boundary between the hydrophilic and the hydrophobic domains that in some areas it is possible for the molecules to protrude across the boundary over the hydrophilic domains, and in some areas reach out or bridge over to another hydrophobic domain. And what is also quite interesting, of these disordered surfaces, the one with the largest hydrophilic and hydrophobic domains with the fewest amount of or length of boundaries, if that makes sense, consistently shows a smaller increase in the overall amount of absorption relative to the other simulations. And this reinforces our belief that this enhancement of absorption is due to patterning effects and the ability of molecules to bridge across and beyond the boundary between the hydrophilic and hydrophobic domains. And moving on to the absorption of hydrophobic surfactants, what we see, first of all, we see the formation of micelles in the bulk. So we have very strong cooperative effects, but also in the simulation, quite slow dynamics. For example, the breakup and the recombination of micelles are generally quite slow. But also, when we look at the absorption process, this proceeds by the absorption of individual micelles onto the surface that then restructures into a more energetically favorable configuration for an absorbed micelle. And because we have the formation of micelles in the bulk, and because we have slow dynamics in the simulation, we will have to say that our sampling in the simulation is perhaps non-ergodic, which means while we do qualitatively see a significant increase in amplification in the percentage increase in absorption of molecules on the surface, quantitatively, there should be quite large error bars associated with the numbers you see for the two rightmost triangles in this slide. Nevertheless, the fact that we do have that considerable increase in the number of absorbed molecules per surface area, even more so than for the predominantly hydrophilic surfactants, means that bridging effects, as I alluded to earlier, are amplified or even stronger for hydrophobic, predominantly hydrophobic surfactants. So in order to interrogate this phenomena further, we examine the simulations on our two hydrophobic stripes. And so the first thing you can see here is the variations in the sizes of the absorbed micelles. So as I mentioned earlier, we have a non-ergodic simulations for the predominantly hydrophobic surfactants. So we do have a stochastic variations in the micelle sizes in the bulk, and this transfers over to a stochastic distribution in size of the micelles as you vary the spatial separation between the attractive hydrophobic stripes. Nevertheless, qualitatively, we do consistently see similar trends. So for example, on the left-hand side, for very close separations, we tend to see predominantly cylindrical absorbed micelle structures, which clearly bridges over the repulsive hydrophilic domain between the two attractive hydrophobic stripes. And on the very right-hand side, we see two very wide separations between the stripes, two disconnected or independent hemicylindrical micelle structures. And then in between those two extremes, we see quite a rich and a complex morphology of both flat and curved bilayer structures. But what is quite clear is the effective interactions of these or length scales of these cooperative effects, which are leading to these different morphologies, clearly extends beyond the length of the individual surfactant molecules themselves, as you can see by comparing the length of this molecule to the spatial separation between the hydrophobic stripes. So another interesting trend we saw when we examined the absorption of different molecules onto the same disordered surfaces was that generally they tended to absorb more or respectively less at the same localized places or locations on the surface, as you can see in this slide. So that gave us an idea. We applied a Gaussian blur algorithm with the expression shown here on the right-hand side with one tunable parameter, sigma, the smoothing parameter, to the nanopattern of the surface itself. And by tuning the sigma value, we were able to fit this Gaussian blur to the spatial density distribution of absorbed molecules that you see in the middle row here. So in effect, we're filtering out low frequency variations in the heterogeneity of the nanopattern on the surface to predict exactly where on the surface these molecules will absorb. So applying this procedure to our striped simulations, we immediately noticed that for larger absorbed structures and examples where we see a greater prevalence of bridging, we get larger fitted values for sigma, which denotes a more long-range effective surface potential in a way. And we applied also the same procedure to our disordered or heterogeneous surfaces, as you can see here. Now, as I mentioned earlier, we do have some stochastic variation for our results, our quantitative results for our predominantly hydrophobic surfactants. So what we did in order to sort of reduce the noise in that data is we averaged, first of all, standardized and then averaged the absorbed molecule distribution for each of the 4 concentrations for each pair of surface and molecule. So what we see is this table here on the left-hand side. And for the predominantly hydrophilic surfactants, we see quite low values for the smoothing parameter, which means that we have a more short-ranged effective surface potential. And conversely, for the predominantly hydrophobic surfactants, we have greater values for sigma, which means we have more long-range effective surface potential. And interestingly as well, if we vary the length of the individual molecules, and we also can see that for the shorter molecules, for example, H3T1, and sigma value is less than, for example, H6T2. And this is simply related to the fact that it's easier for longer molecules to bridge beyond a hydrophobic domain onto another one across the repulsive hydrophilic region in the middle. And finally, if you increase spatial order on our surface, generally what we see then is a decrease in the value for the smoothing parameter, which means that generally we have more short-ranged effective surface potential. And if we have an incoming molecule from the bulk, it will see generally, because there are fewer boundaries, either a fully hydrophobic or fully hydrophilic patch, and therefore the effective surface potential will be relatively flat. So using this information, we can pose a question. Suppose you have the scenario where you have a surface which has a fixed surface composition between two different materials. And the question is, we want to maximize the molecular absorption, but how do we optimize the effective surface potential landscape in order to achieve this objective? Because, for example, if the domains are too small, the attractive wells will be relatively shallow, which will allow the material and the molecules to rapidly detach from the surface. However, if the domains are too large, then the overall surface coverage will be limited to the regions where we have attractive or hydrophobic domains. So in general, we believe that there's a Goldilocks region between the vertices of each of the triangles which you see in this slide, where we get optimal or maximal surfactant absorption. And of course, the precise location of this sweet spot, as it were, will vary from system to system, depending on the inputs and the physical system that it's describing. So to conclude, what we have observed was that for fixed surface area composition, we see more absorption on heterogeneous and disordered surfaces relative to their homogeneous counterparts. Generally, if we increase the molecule length and hydrophobicity, we see an increase in cooperative effects and in the overall number of absorbed molecules per surface area. And when we look at the phenomena of absorbed structures bridging across domains, the effective range of these interactions extends beyond the length of individual surfactants. And finally, this work raises the possibility of tuning surface patterns in order to calibrate the absorption characteristics and properties of various systems for a number of industrial applications. So finally, I'd like to thank my colleagues who worked in this project with me. I'd like to thank Jason from the University of Leeds. I'd also like to thank Sophie from Imperial College London, as well as my colleagues David and Rick from SDFC, and Bill and Michael from IBM Research. Thank you very much for your time, and I look forward to any questions which you may have.
Thank you, Breanndan. I have a question myself. In the beginning, you showed different patterns of the surface that can be generated.
Let me know when I'm at the right slide.
Yes, these patterns. So I just wondered how the structure of this, how this pattern of all these 10 different cases influences adsorption of these surfactants. Is that clear? I mean, if you take, for example, upper left case, you have one pattern, and it's completely different from the lower right case. So if you start adsorbing your surfactants on them, what we are going to see. And what I also notice is that the lower row, it has kind of elongated hydrophilic hydrophobic pattern. So I just wondered how all these cases related to adsorption of these surfactants, and what's the difference in adsorption of the surfactant, how this surface, the structure of the surface influences adsorption.
Well, so what we have in this slide with the 10 disordered surfaces and the relative positions of the surfaces within this triangle is replicated in the results which you see on this slide and on this slide here. So this is the percentage increase in absorption that we get relative to if we did it the exact same simulations on purely homogeneous surfaces. So you can compare the percentage increase, for example, for the two surfaces that you mentioned. You can compare it for in this slide. So, for example, for let's say H6T2, the second one from the left, we have a 14% increase in absorption for that 0,0 top surface which you mentioned. On the bottom right, we have a 6% increase in absorption.
Okay, thank you. And if I look at the same slide, I think you discussed this, but could you remind us this -3 for H3T1 case? So does this mean that we don't observe this enhancement effect that you mentioned?
So I think for I think for that -3, that was comparing it to the homogeneous case. So it's true for that case, this data suggests that we do not see an increase. However, then there is the case that we are running a simulation on a disordered surface generated with these generative probability parameters. So it may be the case that this is a stochastic variation. But I think it's quite clear that the 3% decrease is perhaps not a huge variance overall.
And finally, if you go to the formation of bridges, there is a picture with different, yes, this, for example, the case of 10, the right one. I just wondered, if you add more molecules, would you form a bridge?
That's a very good question. I think eventually, possibly, because I mean, there will be a spatial separation beyond which micelles just will not form at any concentration. But I guess if you keep increasing the concentration of molecules, eventually, I mean, you will have phase transitions in the bulk that might affect it. So I'm not completely sure. It might depend on case-by-case basis.
Okay, thank you. All right. I think we don't have questions anymore.